it depends on the color.
No, this actually.
If it's blue, go look that up.
No, I don't mean the coloration.
Yeah.
But they put the candy coating on the M&Ms.
Yeah.
But they put some type of a coating over the top of it so that the color
Doesn't come off on your hands.
That's
Wax,
why it
probably.
melts in your mouth and not in your hands.
Which they lie.
Where that comes from. You'll go, oh, M&Ms, no thanks.
Blue things. Go check and see. Not right now. Don't do it now. Do it in your free time. But check where different colors come from. I don't have any free time. This is your free time? Chill. Okay, so I sensed a little deer in the headlights look from the last lecture. A lot of that was going on. What is she talking about? Any questions so far on the material? Because, of course, when you got that look on your face, you left class and did what? Went to try and figure it out, right? Or did you just go and have a beer? Don't know what she's talking about. I'm just going to go have myself a beverage. Be done with it. We have to stop doing that, don't we? We have to go find out the answers to our questions. Seek knowledge. So those of you who went to seek knowledge, anybody? Anyone? Good. Make more sense?
I don't understand why. See at the end, like, see where the number 4 is across. Why does it keep going down below stop?
Okay, so the question is, why does that hyperpolarization take place? All right, think about this. Think of those little pumps, those little proteins on the plasma membrane as being paralyzed when they come in contact with a chemical that causes them to open wide. What's sodium going to do at that point? It's going to rush in with its concentration gradient, do what it naturally wants to do. Go from a high concentration to a low concentration. Oh, I'm paralyzed. I have no control over the fact that all this sodium is rushing in as fast as possible, causing what to happen? Depolarization, a change in charge, nerve impulse, action potential, all of that good stuff. When I finally gain control, what am I going to do to get everything back to normal? Repolarization. What am I going to do as a little pump? What would you do if you all of a sudden, all of these things got past you that you were supposed to push to the other side? Once you wake up and get to do your job again, what are you going to do? Shovel all the stuff across where it's supposed to go. Repolarization. You're going to keep doing that really, really fast until, okay, everything equals out. So what happens is a little bit at the end, because I'm doing that so fast at such a high rate, I'm going to kind of overshoot my mark a little bit. Once I figure out, okay, whew, all right, everything is back to normal. I'll pump out at a normal rate. Everything comes back to that minus 70 point. Well, that's the only way I can think of to explain that. So you kind of trying to get everything back to the way it was so fast that you overcompensate a little bit. Make sense? And what's that called? Hyperpolarization. So what happens to the charge? And we're talking about the charge inside the cell. It goes a little bit more negative than minus 70-ish. Again, every single cell has a different set point. That's an ish, an average. Yes? And when does depolarization take place? What point do I have to hit? What's that number called?
The threshold?
It's called threshold. Yes? And for this cell, what is it? Minus 55. Now, is that just, where does that minus 55 have to happen? Just anywhere willy-nilly in the cell? It has to happen at the axon, beginning of the axon, what's it called? The axon hillock, yes? So that wave of change has to come through the dendrites and kind of flow through the cell body region and hit the beginning of the axon. What's going to happen then is something else is going to take charge. No longer is there going to be a change caused by the chemically gated channels, which is what we're talking about in the beginning of this impulse. Who's going to take over at that point? These little channels have different triggers. Who's going to take over at that point? Anyone? The voltage gate. What the heck does that mean? What's going to cause it to become paralyzed and allow sodium to rush in? A change in what? Voltage. Yes? Something came in contact with receptor, chemically gated channel, in the dendrites at the beginning. Whether it be a neurotransmitter, some sort of chemical, or maybe some sort of stimulus. We're going to talk about what we call the special senses next semester. Things like light, or sound waves, or pressure. That's the stimulus that's going to cause this whole thing to happen. But the next guy in line is going to be the voltage-gated channels. And where am I going to find voltage-gated channels? Starting where? The axon hillock, exactly. And running where? Where does the message go? Once it hits the axon hillock, I fire it off, everybody goes crazy. Where does it go? It runs down what? Huh? No. No, you're close still. What's the other one? The axon, yes? When I say nerve, what does that mean? It means a whole group of these cells, yes? The piece. One cell is a what? Neuron. And each neuron has projections. What are they? Dendrites, axons. What's at the end of axons? The terminal end. What happens at the terminal end? Something gets secreted. What might get secreted, you think? A neurotransmitter is very good. Yes. A chemical message that it's then going to send to who? Another neuron or a, what else does the nervous system control? Muscles or what else? Who? Say it loud. No movement. We already talked movement. Think something else. No, that's another neuron. There's three things. Chemicals. What kind of chemicals? Hormones. And who makes hormones? What kind of glands? Endocrine glands. Yes? So that would be the end result. I'm either going to communicate with another neuron through neurotransmitter, or I'm going to communicate with a muscle through neurotransmitter, or I'm going to communicate with some sort of endocrine gland to secrete its product. Okay? And that's going to happen at the other end of the cell. So these diagrams just sort of reiterate the same information. Talks about action potentials, talks about change in charge, and the time it takes for that change to happen down an axon in any given myelinated neuron. No problem. The message happens faster in a myelinated neuron. How come? Because it's insulated. We don't lose any of the charge. It doesn't kind of leak. What if it became unmyelinated? It might trigger other things that don't belong, but most importantly, the message might not, what? Yeah, make it to the end. Yeah, correct. Different diseases called demyelinating diseases are a problem because I can no longer get that message to where I need to get it. Problems with patients with MS. What are some of the problems we see? Problems with moving different muscle groups. Usually the extremities is where we begin. What else? Vision issues. Memory, not so much. But problems with activating different muscle groups. One of the major problems is near the end stages when we have difficulty activating a major muscle group called the diaphragm. And what's that going to cause us not to be able to do? Breathe. Yeah, heart too. So it depends. It depends on the stages. It depends on where the attack is taking place and what it affects on the other end. So there's different ways it's going to manifest itself in different patients. So how long does it take an action potential to run down an axon? A couple of days, 2-3 hours. How long? Milliseconds. Yes? Depending on how long the neuron is, action potentials are going to peak around 2 milliseconds. And by 4 milliseconds, we're almost done, and we haven't hit a second yet. So this stuff happens pretty fast, yes? Okay. Different stimuli is what the next thing discussed in your book is. Propagation of action potential, coding for stimulus intensity, conduction velocity, all of this stuff. Okay. Now, let us not get overwhelmed by all of this stuff. Think about my discussion of hyperpolarization, getting chemistry back to normal. That's important to get the cell back to normal, resting membrane potential. If I keep stimulating over and over and over and over again, am I going to have problems getting chemistry back to normal? Yeah, in the nervous system and in the muscular system too. When we talk about this same concept, when we talk about muscle contraction, we'll talk about repeated stimuli and the chemical effects of that repeated stimuli on those cells. So, can I, if I were to have a whole bunch of dominoes set up and I hit one on one end and they all fell down, is there any way I can make that go faster? The falling down of all the dominoes? Not really. Yeah, even if I hit it a little bit and make them all fall down, they're pretty much all going to fall down at the same rate, right? No matter if I hit the first one really hard or if I just tap it enough to knock everything down. So once I've fired off a neuron, just like those dominoes in a row, and that's going to happen at the same, pretty much same velocity, unless something else changes. So conduction velocity, how fast something happens in the nervous system, depends on the diameter of the axon. As a rule, the larger the axon's diameter, the faster it conducts impulses. The smaller the diameter, the what? How come? Exactly. I have to resist. I have to run along the sides, and that's going to create resistance. Resistance is going to slow things down a little bit. Myelination is also going to be a factor in how fast velocity, conduction velocity. The more that leaks out, the slower things are going to go. So a highly myelinated neuron is going to conduct impulse faster than something that is unmyelinated or not as myelinated. Yes? So they talk about something called saltatory conduction. And that's electrical impulse, what they refer to as leap. It doesn't leap. It gets pushed. It pushes through the center. So think of a neuron axon, myelinated, with those little spaces in between. What are those spaces called again? Nodes of Ranvier. Yes? So we're talking about an axon now. And what am I going to find in those spaces? What I'm going to find is a whole bunch of those little proteins. Voltage-gated channels. Yes? Underneath the myelin sheath, and we're talking about the peripheral nervous system now, do I find a lot of those voltage-gated channels? Not really. So the voltage is going to go in waves down the axon. As the wave sets up, it's going to push through the myelinated area, yes, and smack into a whole bunch more voltage-gated channels, which are then going to do what? Open up and create more sodium into the wave, which is going to swish through the next set of myelin and bang into the next group of voltage-gated channels. You getting the point? So it jumps. It doesn't jump outside. It actually gets pushed in waves through each of the little insulated regions. So that's what that's talking about. Also gives you a little blurb on MS and why that can be a problem. And we see a visual on page 406, figure 11.15, that shows us the action potential waves, the size of voltage. If this particular fiber is what? It's unmyelinated. So what's going to happen to that stimulus size of voltage wave of change as it goes down? Well, it's not the drag, it's the leak, basically. So what happens is we get leakage of some of those ions because it's not insulated, and it kind of peters out. When we have an axon that is able to conduct slow and continuously, it's going to pick up and push along that wave. Even though this happens to be non-myelinated, what we're going to find here is a lot more what? A lot more channels that are going to help to push that along. Excuse me. In a myelinated fiber, what are we going to see? Yeah, that same kind of thing. We increase the amount of sodium every time we hit one of those nodes of Ranvier, and we push through that message at a faster rate. So in a myelinated axon, conduction is fast, and it is saltatory conduction, those little waves that are being pushed along the fiber as we go. I have no idea. Myelinated fibers, we find lots of them in the peripheral nervous system. Central nerve, non-myelinated, typically we find those in the spinal cord. Short distances they have to travel. Interneurons, some of those are non-myelinated. Yeah, interneurons. What's an interneuron? Do we remember? Did we talk about that yet? Your kind of hands-off message between motor and sensory information. We're going to find those in the spinal cord. When we go and we look at the spinal cord and the brain proper, there's two different areas of tissue we're going to discuss. One of those areas is called white matter, and that is where we're going to find myelinated fibers, many of them axons. The other portion of both of these in the central nervous system is gray matter, and that's where we're going to find cell bodies and non-myelinated fibers. Yeah, and typically very short distance has to travel, as opposed to a myelinated fiber, which tends to be a longer distance. Exactly. When I look at the sciatic nerve, I'm going to see neurons in there, and we described them last class, where the cell bodies are way up around the lumbar region, and then the end of those fibers are way down in your heel. So big, huge, long. We have to make sure we insulate that so that we don't lose the message being sent. Okay? So do you understand the difference between a myelinated and unmyelinated fiber? There's different groups of fibers, group A fibers, group B fibers, and group C fibers. They have different diameters. Some of them are myelinated, some of them are non-myelinated. Just a basic definition of the different fibers on page 406 in your textbook. Okay, so this is one neuron talking to another neuron. Are neurons all glued together? No. There's a space between one cell and the next. Oh, there's a fancy name for that space. Do you know what it is? It's called a synapse. Okay? And the space in the synapse can be also referred to as the cleft or synaptic cleft. But the synapse is a space between the end of one neuron and something else that it might communicate with. Now, what might it be communicating with? Endocrine gland, muscles, or other neurons. Exactly. In this case, who are these? What's this interaction I see here? Yeah, another neuron. So there's different synapses, different spaces between one neuron and its effector. Presynaptic would be before the synapse, before the space. Synaptic would be what? After the space. Now, there's different messages that any given neuron can give off that will fire off something postsynaptic. There can be electrical synapses where there's a change in charge that flows in that space like a wave. Remember, all of this stuff is happening in fluid. Just look, if I was to take a beaker of water and put a drop of dye in it, it would diffuse through the solution. These changes, these chemicals, these electrical changes, when I'm adding them one place, they diffuse through the solution. They go from a high concentration to a low concentration, just like that dye would in your beaker of water. So when I start or set up an electrical change, what kind of receptors are going to be affected by that? Is a chemically gated receptor going to be affected by a change in charge? It would be a voltage-gated receptor. So different receptors are affected by electrical changes, changes in charge, changes in flow of ion concentration. The other type of synapse we find is something called a chemical synapse. And what might the receptor on the other side of that space be affected by? chemicals, exactly. Changes in some sort of chemistry. The receptors are typically receptive to chemicals called neurotransmitters. And they flow through the space between the end presynaptic neuron of one neuron. And what's at the end of the neuron? What's that place called? Well, the cleft is a space. Say it again. The neurotransmitter is what's going to flow through the space. What's the end of the axon called? Yeah, the axon terminal. Sometimes you'll see it referred to as the axon end bulb. So information is going to be transmitted from one neuron to the next. Now, that information in this case is in the form of neurotransmitter, a chemical. Well, what causes the chemical to be released? Any ideas? Exocytosis, very good. But what causes exocytosis to happen in that neuron? This is the presynaptic one. That's the postsynaptic one. Something happened. What? What kind of stimulus? No? Yeah! In the form of a what? We've been talking about it the whole time. Yeah, action potential. So when a neuron, presynaptic, fires off, that change in charge that spreads through the axon, its end result is what? Reaches the end of the axon and causes what to happen? Exocytosis of what? Chemicals. What are we going to call them? Neurotransmitters. So in this diagram, you can find it on page 409 in your textbook, we see that axon terminal, just one of them. Are all axons of all neurons, do they only have one terminal? No, they kind of flare out at the end and have several. So we're just looking at one terminal end here and one synapse. So when that change in charge, the action potential, arrives at the axon terminal, there's going to be a shift that occurs. One of the things that happens here is that change in charge is going to affect the little calcium channels that are on the plasma membrane of the axon terminals. Kind of the same thing that happened when sodium rushed in. What's going to happen with calcium here? It's going to rush in too, exactly. And the rush in of calcium is going to cause a change in charge at the synapse, or the, axon terminal. That change, increase in calcium, is going to cause the axon terminal to do what? Who? Release what? No. You're on the right track, but it's going to release. Look at the picture. What's it going to release? Those little things with the little green balls in them, right? What's the little things with the little green balls in them? Yeah, they're little vacuoles that are filled with, what's the little green balls? Neurotransmitter. Yes? They're going to spit out into the fluid. What's going to happen next? Those little, whatever they are, neurotransmitters are going to go from a what to a who? High concentration to low concentration across the space. Yes? And they're eventually going to come in contact with who? the little receptor on the other end. They call me a receptor if I'm affected by the chemical. So that particular neurotransmitter, whatever those little green balls are, when they come in contact with a receptor, a little protein on the surface of the plasma. Now, what the heck is this thing down here? What is that? It's another neuron. When we come in contact with that neurotransmitter comes in contact with the receptors on that other neuron, it's going to cause what to happen? Same thing we've just been talking about the whole time. Sodium is going to then do what? Rush in. So depending on how many of these guys I fire off, that's going to tell me whether or not I'm going to have an action potential in the postsynaptic neuron. Yes? If I only have a few little neurons that fire off and tell it to move, is anything going to happen? Probably not. What do I have to do in order to have an action potential happen in the postsynaptic neuron? Say it out loud. You have to reach threshold, exactly. You have to make enough of a change to change the charge. Where? Axon hillock. Yes? Once that neurotransmitter has been released, the cell, presynaptic neuron, is going to go back to normal. Yes? Just like all the other chemistry, just like depolarization, repolarization, our discussion before. That neurotransmitter that hangs out inside that synapse, is it going to work forever and ever and ever? What happens to it? Almost immediately this happens. What's going to happen to it? It's either going to diffuse away and be too low in concentration to really have any effect on anything, or there's going to be specific enzymes that are going to do what to it? Break it up, break it down, break it apart, so that it doesn't continue to cause an effect. How am I going to fire off that postsynaptic neuron again? What do I have to do in order to fire that off? I got to fire that guy off. Yes? I have to create more of a stimulus in order for the postsynaptic neuron to be fired off continuously. Does that make sense? So, information transfer across a chemical synapse might make a lovely little essay question for your next exam. You can see its explanation beginning on page 408. The diagram on page 409 outlines it for you visually, and we continue the explanation on page 410. Now there's a tiny little bit of delay, and I'm talking very, very tiny little bit of delay between presynaptic and postsynaptic excitation. What's the delay? Why is there a delay? What has to happen? The neurotransmitters need to be released, and they need to diffuse across the space. They need to come in contact with the receptors on the postsynaptic neuron, and then we have to create that chemical wave before we fire off that postsynaptic neuron. So what's the big, huge delay here typically? It depends. 0.3 to 5 milliseconds, depending on the different types of synapses. Okay? So understand what happens at a synapse. Understand there's different types of synapses. There's different stimulus. Again, when we talk special senses, it's not always going to be a neurotransmitter that fires off a neuron. Right? I just fired off a whole bunch of neurons. Do you know how I did it? I scared the booyah out of you, didn't I? But how did I do that? I just put forth a stimulus. What was the stimulus? Sound. It's a mechanical stimulus. Every time I touch something, that's a stimulus. It's not a chemical, is it? It's a what? It's pressure. It's mechanical. So we're going to see that there's different types of receptors that will respond to different types of stimulus. So in my ear, I have mechanoreceptors that are stimulated by a mechanical change, actual physical movement. If I scream into your eyeballs, will you see better? Not really. I might open my eyes a little bit like, what's going on? But I'm not going to see better because what type of receptors do I have in my eye? They're stimulated by light waves. They're called photoreceptors. Yes? So we'll see that there's different types of stimuli that will fire off certain types of neurons. Each neuron is singular. It can only go in one direction, and it can only be fired off by very specific stimulus. Again, if I scream into your eyeballs, you're not going to see better. If I shine a light into your ear, you're not going to hear anything. Yes? Right? Because those are specific stimuli that fire off those specific neurons. Not all neurons are going to cause excitation. Some neurons are actually going to stop things from happening. So the next part of the book talks about inhibitory or excitatory postsynaptic neurons. So excitatory synapses, or EPSPs, are going to do what? Are they going to cause depolarization? They're going to help bring neurons closer to what? Threshold, and threshold is then going to cause a neuron to undergo what? Depolarization. What's another name for depolarization and repolarization and all that stuff? Action potential. Inhibitory is actually going to cause binding of neurotransmitters. Inhibit sodium from rushing in and stopping what from happening? Yeah, no more depolarization for you, okay? So we're going to stop neurons from firing off. We'll see when we talk about controls for your respiration. Breathing rate. That's all controlled by who? Who controls your breathing? Central nervous system, yes? And we're constantly being told, breathe in, breathe out, breathe in, breathe out. What if I have to slow it down? How do I do that? If somebody's always telling me, breathe in, breathe out, breathe in, breathe out, breathe in, breathe out, who's going to slow it down? It's the portion of the nervous system that's actually going to slow down some of those nerve impulses. We'll talk about next semester in the respiratory system areas called the pneumotaxic and apneustic regions that work hand in hand to control your rate of breathing. It's going to slow down the breathe in, breathe out message when we have to slow down our breathing rate. Why do you breathe? To live. Why? Correct. Now, I have to breathe in, gas exchange has to take place, oxygen, carbon dioxide, and then I breathe out waste gases, right? When would I need to slow down or speed up my breathing? How would I know to do that? Balance of what? Or respiratory gases, right? So I need a certain balance. Just like everything else in the body, I need a certain amount of oxygen and carbon dioxide balance. Yes? But why would you speed up breathing? And this is my, yeah, because you'd have to run to the bookstore to buy Hermits, because they sell those sometimes and I really like them. Why would my breathing rate increase when I had to run to the bookstore to buy my Hermits? Because you're contracting muscles, you're using up more oxygen, your central nervous system is detecting what? Oxygen levels are going down, carbon dioxide levels are going up, increase my breathing rate. But what happens once I get my delicious cookies and I sit down to enjoy them? I'm not contracting as many muscles anymore except in my mouth and my digestive system because I'm eating them, right? So what happens to those levels? You see where I'm going with this? So carbon dioxide and oxygen levels in the body are going to dictate breathing rate by communicating nervous system communication with the respiratory system. Yes? When those levels start to become unbalanced, or one of those little seesaw things comes into play, homeostasis and all that stuff, that's when the nervous system is either going to speed up or slow down. If I slow down my breathing rate, and we're going to see when we discuss the respiratory system, even though carbon dioxide is a waste product, it's going to help control chemical balance in our body and pH. So we have to maintain a certain level of both in order to maintain the proper chemistry of metabolism in our bodies. So if we're breathing too fast, anybody ever hyperventilate? Get nervous sometimes, or just get so exhausted, tired that you start breathing too fast and you feel like you're going to pass out and get sick? What do they have you do? They grab a paper bag and say, breathe in, baby, and slow down. Why do they make you do that? Huh? You're actually getting rid of your waste too fast. So what they have you do is breathe back in the air that you breathe out, which is at a little bit higher a concentration of carbon dioxide than it is oxygen, to help bring carbon dioxide levels back to normal. If you breathe out too much carbon dioxide, your blood pH is going to be affected by that. Yes? You're a fine chemical balance. And who controls it all? Many of these cells, in some way, either directly or indirectly, are going to control that chemical balance. Amazing, isn't it? So, excitatory synapses, inhibitory synapses. And because any given neuron can communicate with more than one postsynaptic neuron, we might have a summation of several different neurons for any given message being sent out. So they talk about different summations: spatial summation, temporal summation, excitatory, and inhibitory. Basic definitions of those guys. We'll see them again when we discuss other parts of the nervous system. We'll see them again when we discuss other systems and how the nervous system affects them, just like our discussion right now on the respiratory system. No, I forgot to make the sign-up sheet. You get a freebie today. So, on page 412, table 11.2, we see a comparison of some of the different action potentials. When we talk about stimulus, we always have to keep in mind change in charge Reaching where? What part of the neuron are we concerned with that change in charge reaching? The axon hillock. So if I start my message coming from the very, very ends, the dendrites, it's going to take me a little bit longer to create that sodium wave that reaches the axon hillock. So they talk about some of the different distances this change has to travel. If my synapse is very close to the axon hillock, that will happen much more quickly than if my synapse is way out at the dendrites. Does that make sense? Because I have to cause a change in charge enough to reach threshold and enough to fire off my what? What's in the axon? Yep, what kind of channels? The voltage-gated channels. Did you have a question, hon? Not necessarily connect, but where the message is going to affect. It could be in the dendrites. It could be closer to the axon hillock. It could be on the cell body. If you look at this neuron, you see? Where is the connection? Where is it's closer to the cell body. Is that going to take less time than something that might connect out at the dendrites? Yes. What if that connection was right around the axon hillock? See that blue one? So there's different meeting places. That one is called an axonal synapse because, where is it? It's the axon of one and the axon of another. What's the other one called? The axosomatic synapse. Soma means what? Body. So the connection is where? At the cell body. So again, they're very different. Usually the ones that have to happen the fastest typically hang out around the axonal synapse region. You're welcome. All right, so distance traveled, short distances versus long distances are discussed here. Different types of stimulus. Repolarization, summation, some of the terms we want general ideas for. So temporal, spatial summation are discussed on page 412. Basic definitions of those two terms is what I would like you to understand. Excitatory and inhibitory. Oh, I'm going to go back one. Some of the differences between those. Typically, the initial effect of a stimulus is to change ion concentration. Typically, that ion that we're going to discuss is what? When we start the beginning of an action potential, what ion is going to rush in and cause the whole business to change? Sodium. At the end, what are we talking about? What's going to cause those little vesicles of neurotransmitter to be secreted? Calcium. Okay? So sodium in the beginning, calcium at the end. These chemicals, neurotransmitters, aren't all the same. The next portion of your book talks about neurotransmitters and their receptors. One of the most popular ones is something called acetylcholine. And when we talk about acetylcholine, we'll see it affects widespread throughout. It's in the central nervous system, it's in the cortex, it's in the hippocampus, it's in the brainstem. Those are all part of the brain proper. It's in the peripheral nervous system. All neuromuscular junctions with skeletal muscle. This is going to be the chemical message. So when we hit the muscular system at the end of first semester and we talk about an action potential, we're going to see a lot of the same terms we just discussed in the nervous system. One of the reasons we kind of flip-flop the chapters, because when we discuss the neuromuscular junction and a nerve impulse and an action potential, everybody looks at me like I have 14 heads because we haven't discussed it yet. Now we will have discussed it. So when we hit that part and we look at a muscle contraction and we look at a tracing of a muscle contraction, what's it going to look exactly like? The tracing of a nerve impulse. And the terms are going to be exactly the same. Action potential, depolarization, repolarization. That's why we learn it first before we discuss it in muscles. So when we talk about the chemical that's going to cause that change, cause a chemical change within a muscle to cause it to change, lengthen to shorten, we're going to talk about this guy, acetylcholine. But that's not the only one. There's others. This table, 11.3 on page 415, gives us a list of some of the different neurotransmitters and their effects. Basically, I want you to understand that there are different ones. You should recognize acetylcholine because that's the big player. We're going to see it referred to as ACH a lot. So the next time we discuss a nerve impulse, usually you're going to see ACH in there, and you'll know that's a neurotransmitter. What breaks down acetylcholine in the synapse? Big long word. Well, look at the end of the big long word. It's an enzyme. How'd you know that, smarty pants? ACE. A-S-E. Acetylcholine esterase is the enzyme that's going to break down acetylcholine in the synapse. Remember, we just talked about enzymes breaking down neurotransmitter. I see it on page 414 under the description of acetylcholine. See that big bolded word smack dab in the middle of that paragraph? So that's the enzyme that's going to break down that neurotransmitter. The next group of neurotransmitters we're going to see are things called biogenic amines. And we're going to find those scattered in different places throughout. Some of them are only in the central nervous system. Some of them are rare in the peripheral nervous system. Some of them are going to hang out in very specific regions of the brain in the central nervous system. Please just recognize them as the group that they belong to. So biogenic amines are norepinephrine, dopamine, serotonin, and histamines. Some neurotransmitters are protein in nature, amino acids. They start on page 415 and they go over to page 416. Again, glycine, glutamate, and the one on page 415 is called gamma aminobutyric acid, or GABA. Just understand that amino acids can also act as neurotransmitters. Again, if you look at the description, typically we're going to find these where? What part of the nervous system? The central nervous system. Again, other protein in nature peptides are listed here. More central nervous system. We will see some of them when we discuss the digestive system. Somatostatin and cholecystokinin, CCK, are associated with the digestive process. But most of the other guys are central nervous system again. Purines. What's a purine? Any clue? You heard that word before. You heard that word in chapter 2. No? Yeah, ATP. What organic molecule family does it belong to? Amino acids? No. Who? Somebody said it? Nucleic acids, yes? Purines. Adenine. Guanine. Pyrimidines. Thymine. Cytosine. Do you see where I'm going with that? So those can act as neurotransmitters as well. Again, very rare, but we see them central nervous system. In peripheral nervous system, very close to the spinal cord. And then some other gases and lipids can also act as neurotransmitters as well. Nitric oxide, not nitrous oxide, nitric oxide. What's nitrous oxide? The stuff they give you at the dentist. What do they call that stuff? Laughing gas, exactly. Different. Brain, typically brain, a little bit in the spinal cord. Carbon monoxide can be excitatory or inhibitory, or it can kill you. Yes? So it can affect neurotransmission in the brain. So just a general idea of these guys. Classification of neurotransmitters by function. Excitatory versus inhibitory. Direct action versus indirect action. Basic understanding of the fact that not all portions of the nervous system cause a direct effect. They might cause an indirect effect. And that has to do with some of the different neurotransmitters that are being released from those neurons at any given time. I'm not going to get too crazy about that. What is this? Yep. So the normal brain, what are all those colors? Activity, yes. Actually, what kind of, what am I seeing? When the colors are bright, yeah, I see release of chemicals, I see nerve transmission, I see activity going on. Abuser. Cocaine abuser, different? What's not going on? Not much at all, yeah. But see what happens after we have discontinued our use? What's going to happen? Well, it's killed a lot of stuff, but 100 days after cocaine use? Yeah, but what are we doing little by little after we get rid of, activity does increase. So that's kind of interesting if you're interested in the effects of chemicals, including drugs, on brain activity. Remember, you kill it, you don't get it back, right? We use very little of our brain, actually. We have a lot of file cabinets upstairs that are empty. So we have the ability to kind of spread out and use other file cabinets, but the more file cabinets we destroy, we will never regain them. And when I say file cabinets, do you understand what I mean? Yes. Groups of cells in the brain that store information for us. So drug use, not really a good idea. And quite expensive, actually. And cause a whole lot of other problems in our lives besides brain function. So. It not regenerate new cells, regenerate new connections. So you know my axons that have all those connections? I can make more connections. But once I kill the cell body of a cell, I'm not getting another one back. Make sense? All right, so Direct neurotransmitter receptor mechanism, channel-linked receptors are going to have a direct effect on ion flow and a direct effect on sodium rushing in and a direct effect on a change in charge. But not everything is always direct, of course. The other type of change that can happen within a cell is an indirect change. We call this a second messenger system. When we discuss hormones second semester, we're going to talk about water-soluble hormones and this chain reaction of events that happens within the cell when a receptor comes in contact with a chemical. In this case, because we're talking about the nervous system, that chemical that comes in contact, and we can see it way over there on the other side of the diagram, the little red ball coming in contact with that purple thing, a receptor, is not going to cause something to flow into the cell, but it's going to cause an internal change within the cell. Very similar to what that inflow of sodium did. In this case, this is called a second messenger system. And it involves kind of an intermediate guy that's going to pass that change in charge, change in whatever, across the inside of a membrane. That passing of change is going to cause the cell to do something different, metabolize differently, have a different chemistry. So, we talked a little bit about second messenger systems in Chapter 3. You remember that? We saw these little guys running across the top in Chapter 3. But the receptor for any given neurotransmitter would be on the surface of the cell. And when that neurotransmitter came in contact with the receptor, it would then set off a chain of events within the cell to cause it to do something different. In this case, the ligand, the cute little ball in the road race we see at the top, would be our neurotransmitter. It would come in contact with the receptor, that little protein on the surface of the cell, and that would set the chain of events going. It would cause a change in shape, which would then kick off a G protein. That G protein would then start moving along the inner surface of the cell and come in contact with some sort of enzyme. In this particular second messenger system, we see it come in contact with an enzyme called adenylate cyclase. When the two bang into each other, it causes the formation of a second messenger, something called cyclic AMP. And we see it over there running across the top as well. The presence of that molecule is going to cause changes to occur in different proteins within the cell. In this case, it's going to take an inactive protein or enzyme, and when it bumps into it, it's going to activate it and cause the cell to do something different. So through second messenger systems, we can cause changes within the cell that will eventually do what to those ion channels? Open them up. And when they open up, what's going to happen? Sodium's going to rush in. And then what's going to happen? Depolarization and the whole action potential thing. So this is an indirect way. The one we just looked at before is much faster, yes? In a direct way. Many of your hormones work this way. So they will cause changes indirectly through second messenger systems. Yes? You with me? So presynaptic versus postsynaptic. Remember that synapse or the fiber can branch off in many different directions and create all kinds of different circuits to fire off in different patterns. And that's what the next portion of your textbook talks about. So basic concepts of neural integration. One neuron firing off many other neurons. One of the examples I use is my book bag. Now right now my computer's out of it, my book is out of it. So when I pick it up, I only have to contract a certain number of muscle fibers to create enough of a pull to lift it. But what happens when I put my book back and I put my computer back and all the plugs back and my keys back? The same action has to take place, but in order for me to lift this bag, what do I have to do? I have to contract more fibers, more muscle fibers, shorten more of them to create more of a pull. You with me? So that's what this is sort of talking about. Depending on what has to happen, I might have to initiate more stimulus from the central nervous system to more effectors. In this case, it would be my muscles to pick this bag up, depending on what I have to do. So that's basically what they're talking about in this section of the book when they talk about the organization of neurons, different types of circuits, patterns of processing, and we see that the neurons communicate in several different ways to several different groups of other neurons. So a basic understanding of that is all I want you to have. You with me? So again, figure 11.23 on page 422. Just a basic understanding of some of the different circuits created in the nervous system. What happened? What'd you do? Did you think about what you just did? You just did it, right? Some of you who are watching me are like, she's losing it. But some of you who are like spacing out already just got the bejesus scared out of you, didn't you? You just experienced something called a reflex arc. You experience this when you put the pot of water on the stove and then you go to grab it and you forget, oh, that's hot. And you pull your hand away before you get the chance to think what you're doing. What we're doing is sending message that sort of gets sent or handed off right at the spinal cord area to protect us from damage. When you hear a loud bang and sound, what do? You quickly, a lot of us will close our eyes and move our head out of the way. Why? You're protecting yourself from impending danger, right? When you pull your hand away from that hot pot, what are you doing? Protecting your skin cells from further damage. If I had to stop and think about it, more damage would take place, right? The next part of the book talks about a simple reflex arc. And when we talk about the following chapter, when we discuss the following chapters, we'll get into that more in more detail. But reflexes are patterns in information coming in and information going out. They cause the motor information to happen at a much faster rate. Protection mechanisms for us. These are the components of a simple reflex arc. So all reflex message has the same basic components. So there's some sort of message coming in, and it's being received by what? A receptor. So in the case of me dropping my book, I dropped it, it hit the deck hard. The receptors were going to be found where? If you weren't looking at me. In your ears. They're going to then send message where? Central nervous system, right? Through what type of neuron? A sensory neuron. Remember, I can only have one type of stimulus and I can only go in one direction. So information coming into the central nervous system is going to be sent by what type of neuron? Sensory neurons. That information is then typically handed off by an integration neuron or an integration center. In this case, it's that interneuron, the green one, Typically, it's sent to the spinal, well, it's in the spinal cord. Sensory information will hand off to interneuron, and then that interneuron will send information up to the brain. You'll process that information, and then you'll send a message back down the spinal cord and out to cause an effect. If it took me that long to do that, I would burn the living daylights out of my hand when I went to pick the pot off the stove. So in a reflex arc, I'm going to hand off, interneuron is going to hand off immediately to who? To a motor neuron. I'm going to send that message right out to cause an effect. In the case of me slamming the book, the effect was close my eyelids. And what's going to cause me to close my eyelids? Some sort of motor neuron that's going to cause an effect. Pull my head away. How do I do that? I have to contract a bunch of muscles to do that, right? Who's telling me to do that? Motor neurons. I have to pull my hand away from the stove. How do I do that? Motor neurons causing an effect in muscle groups. So motor neurons are then going to send message out and cause an effect, some sort of response. In the interim, though, that interneuron does what it's supposed to do normally. And it also sends message up the spinal cord for processing. So by the time you've already pulled your hand away from the stove or blinked your eyes and pulled your head away, you now actually are aware of what just happened. Correct. You pull your hand away and you're like, oh shit, that's hot. Yes? But you do the, oh shit, it's hot after your hand is out of there. You see where I'm going? So that's what a reflex allows for. Protection for you. Protects you from more damage that could potentially happen. And reflexes we'll discuss in the next chapters. So you should understand the basic components of a simple reflex arc. Developmental aspects of neurons, don't worry about it. And yes, my friends, we are done with chapter 11. How exciting! So here's the drill. Chapter 12, chapter 13, chapter 14. If you haven't had a peek at them yet, just take a little flip through. They'll make you nervous as hell. But here's the deal. You have tools in your hands as we speak. And I am going to follow them to the T. Do you know what those tools are? The chapter outlines. Where are they? Online. CM Connect. I will follow the chapter outlines to a T. When I create my exam for the nervous system, which will be chapter 11, chapter 12, chapter 13, chapter 14, where am I going to get the questions from? You got it. Yes? So I will open up the quiz for chapter 11 this afternoon sometime.